Hey guys, my name is Abhishek and welcome back to MBBS Treasure Channel. Today we are going to talk about the mediators of inflammation. Till now we have talked about the inflammation and its three major clinical phases, that are alteration, exudation and proliferation. Let's now read about the mediators that are formed after the process of alterations. And in the alteration, what happens, there are two phases like primary alteration and secondary alterations. So let's see how the mediators are going to make the self injury, that is otherwise known as secondary alteration. Okay, so let's start our today's video. So friends, till now we have learned that the alteration method, it means the primary injury that will be caused by the etiological factors, and that is going to show the manifestation of inflammations. Next, what happens after the primary alterations, there will be secondary alteration. It means the self injury that will be formed by the mediators. Okay, so now let's see that what are the mediators of inflammation. So now we are talking about acute inflammations that will be occurring within a very short span of time. Okay, and the inflammations are also divided according to acute and chronic. So now we are going to talk about the mediators of acute inflammations. The mediators of acute inflammations are divided to cellular mediators and blood plasma mediators. As you can see the diagram over here, the cellular mediators are produced from the cells, and these cellular mediators are being divided according to the formation process of the mediators, whether it is pre-formed, whether it is formed beforehand and it is well prepared for the situation, or it will be produced at the very time or at the point of inflammation or at the point of injury. Okay, so according to the time, it can be divided according to the preformed mediators and newly synthesized mediators, and these mediators will be formed from the cell itself. Okay, so the red color that I have shown, it is already preformed, as it will be present within a vesicle, and when the situation will comes or when the injury will takes place, then these preformed mediators will get released at the site of injury, and they will cause the first response of the inflammations, like there will be vasodilation, increasing in the vessel permeability, increasing in the blood, that is hyperemia condition. So these all will be formed, or these action will be performed by these preformed mediators. What are these preformed mediators, and which cells are going to produce these kinds of preformed mediators? Yes, the preformed mediators are nothing but the histamine, serotonin and the lysosomal enzymes. Okay, and these preformed enzymes are going to be produced from where? These are going to be produced from the cells below the site of injury, and these cells are nothing but the mast cell, basophil cells and the platelets, neutrophils and macrophages. These mast cells and macrophage cells are already present at the site of injury, or it is present at the tissues, but these basophils and blood platelets, neutrophils, these are blood cells. They will be coming to the site of injury and they will be performing the release process of these preformed mediators. Okay, so these preformed mediators are like histamine, serotonin and lysosomal enzymes. Histamine and serotonin both have the function of increasing the vessel permeability, increasing the they will be increasing the content of blood at the site of injury, that is hyperemia, and they are going to dilate the vessels. First, when the injury will takes place, for the very first instance there will be vasoconstriction, but after certain time when the histamine will get released, then the blood amount to the region of injury will increase, and that is performed by this histamine and serotonin. Next comes the lysosomal enzymes. Lysosomal enzymes can be two types. It can be primary and secondary enzymes. These lysosomal enzymes, actually what it helps in breakdown, because these lysosomes have the hydrolytic enzymes. Okay, and it has a special enzyme, that is phospholipase A2. And what is the function of phospholipase A2? We are going to discuss discuss it in a very brief manner in the arachidonic acid metabolites, because for the beginning of the process of arachidonic acid metabolism, we require a phospholipase enzyme A2 that is a part of this lysosomal enzyme secondary. Okay, now let's talk about the newly synthesized mediators. What are these mediators? These can be arachidonic acid metabolites like prostaglandin and leukotrienes. It can be nitric oxide. It will be produced by the which cells? It will be produced by the endothelial cells of the vessels, and it can be cytokines, interleukins. Cytokines and interleukins are mostly produced by the leukocytes, and platelet activating factor will be activating the platelets. So these are the arachidonic acid metabolism, nitric oxide. So these are the newly synthesized because they will be formed after the process of injury. Rather, these preformed mediators were formed before or earlier, and they were responsible for immediate cause of actions. But these will be formed after 5 to 10 minutes of injury. For this process, we require time. The most important part of this newly synthesized that we should know that it should be arachidonic acid metabolism and cytokines and interleukins. That we will be having a separate video for its discussion, because this arachidonic acid metabolism is very important, because this concept will help you for any kind of inflammation, any kind of disease, because this arachidonic acid is nothing but a part of cell membrane, and when the cell membrane get destroyed, then this arachidonic acid metabolism will be starting, and it will be producing the mediators like prostaglandins and leukotrienes. And these prostaglandins and leukotrienes are going to have different kinds of effect on our body that needs a special attention. Okay, and these cytokines and interleukins too. So for now, you should know that we need to classify the mediators according to preformed and newly synthesized. So next comes the blood plasma mediators. These blood plasma mediators are being divided according to complement system and the factor 8 Hegman's factor activations. These blood plasma proteins, as we know, the blood plasma proteins are nothing but these are the proteins that are derived from the liver, and the liver is the main source for the production of these kinds of proteins, whether it is complement system, whether it is activating for the process of coagulations. So this liver is responsible for productions of these kinds of mediators. When there will be activation of factor 12, I want to make sure that this factor 12, you should remember that after when the inflammation takes place, first the factor 12 is going to be activated. It's a special name that's given under the scientist name. It is Hegman factor. Okay, this factor 12 is going to be activated, and what happens upon the activation of factor 12? The plasminogen that are freely present in the blood vessels is going to convert into the plasmin protein, and this plasmin protein is going to activate this complement system. So when the factor 12 is going to be activated, then it will be further going to activate the complement systems. And as we have already read about the complement systems in the hypersensitivity reactions, like there can be C3A, C5A, C3B and C5, 6, 5B, 6, 7, 8, 9. This is nothing but the membrane attack complex. So alternatively, what is going to happen? This factor 12 is going to activate the plasminogen to plasmin. It will help in the process of conversion, as it will act as a catalyst. Then what happens after the activations of the factor 12 itself? It's going to activate the further kinase system. Kinase system is nothing but the production or the process of production of kinase. This also requires a special video, and I will be mentioning these three important process, the arachidonic acid, cytokines, interleukins and the kinase system, in a separate video. Okay, so for now you should know that kinase system will be activated and it will be producing the kinase. Why this kinase is helpful? Because the kinase will produce the vasodilation, it will increase the secretion of the fluids at the site of injury, then it will increase the process of pain, means it will increase the reception process of nociceptors, and it will cause the pain. Next comes the coagulation system. As the site is already injured and there is hemorrhage at that site, means blood hemorrhage, and the blood is bleeding. So what will happen? This coagulation system is needed to be activated at that site. That is the reason why the factor 12 gets activated, and then it activates the coagulation system, and that leads to clot formation. Next, what happens? This fibrinolysis system. This is a very bad system that gets activated upon the activation of this factor 12, because this fibrinolysis will break down the fibrins at that site of injury. But it is also helpful because it helps in the eradication or elimination of the necrotic fibrin molecules, because they will be producing the fibrin degraded products. Now let's come to complement system. What happens upon the activation of complement system? We know that complement systems are a group of proteins that will be helping towards giving response to certain kind of infections or any kind of bacterial or microphysic infections, so that our body can build the immune response towards the causing factors. That is the reason why this complement system will be also activated at the point of inflammations. So what are the complement systems that are going to be activated? It will be C3A and C5A. Especially these two have special name, that is anaphylatoxins. Why they are known as anaphylatoxins? Because they are the toxins that will be producing the anaphylactic reaction. And anaphylactic reaction is nothing but the type 1 hypersensitivity reaction. Then next, C3B. As we have read that C3B acts as an opsonin in the type 2 as well as in type 3 hypersensitivity reactions. So they are going to increase the process of phagocytosis of the bacterial products or the patterns that we have already read. Next comes the MAC molecule, or the membrane attack complex. What is the function of membrane attack complex? It is going to produce a enzyme, that is perforin, that is going to perforate the cell membrane of the bacteria. Okay, so they are ultimately going to make the lysis process and phagocytosis process that will be helping the body to defend against the bacteria or any kind of infections. So the complement system also gets activated through this mediator process. Now there are some also factors like negatively charged surfaces of basal membrane of the vessels, collagens, elastins and glycosaminoglycans. Yes, these are the structures that are getting destroyed at the site of injury, and they will be gaining or they will be inducing the negative charge effect. And the bacterial lipopolysaccharides or the enzymes like trypsin and plasmin, they all are going to make or stimulate the increasing in the blood plasma mediators. Okay, so these are also the key features for increasing or stimulating the process of inflammation.